No, thanks very much for inviting me and to basically share the exciting news of the latest data from the BASIS clinical trial. So the BASIS clinical trial was a phase 3 open label, multicenter, one-way crossover clinical trial. The individuals from the study design perspective remained on the standard of care for a period of 6 months, after which they then switched to prophylaxis with Marstacimab for a period of 12 months. In the latest clinical trial, which involved those with inhibitors, in fact, the study design was exactly the same. 6 months of observation during which they received their standard of care, and then 6 months on Marstacimab. The dose of Marstacimab that was used was a loading dose of 300 milligrams given once, followed by a weekly subcutaneous dose of 150 milligrams of Marstacimab, and that continued for a period of 12 months. The results, in fact, were almost similar to the results that we saw during the BASIS clinical trial without inhibitors. Essentially, if one looks at the primary endpoint, which is the annualized bleed rate, there was a drop in the annualized bleed rate from an ABR of approximately 20 to an ABR of less than 2, clearly demonstrating superiority of Marstacimab prophylaxis over on-demand treatment. And in fact, if one specifically looked at the proportion of individuals that did not bleed at all whilst they were receiving Marstacimab, that number, in fact, was in excess of 50%. So from the safety perspective, Marstacimab during the exposure period of 12 months did not result in any serious adverse events of note. In particular, there were no thromboembolic events. There were certainly no thromboembolic events and thrombosis that were directly linked to Marstacimab. Some individuals developed anti-Marstacimab antibodies. Those did not have any impact on both the pharmacokinetic as well as the clinical features resulting, for example, in excessive bleeding in these patients. So overall, if one looks specifically at the inhibitor cohort, the results, in fact, were consistent with what we saw in the non-inhibitor cohort using the same dosing regimen of Marstacimab. No, thank you for asking. And I must perhaps preface the answer to your question by expressing our excitement in living in this era of unprecedented exponential increase in the therapeutic landscape in hemophilia. In particular, we are excited about drugs such as Marstacimab, which obviously enrich our therapeutic toolbox. So the implication for the hemophilia A and B with and without inhibitors, in fact, is huge. What it does, what Marstacimab does, is that it increases the breadth with which one is able to tackle hemophilia in general. As you know, it works independent of factor 8 or factor 9, and in fact, it works independent of the inhibitor status. So it can be used for both hemophilia A and B, unlike other non-factor therapies, which may be limited to one phenotype. This particular one, in fact, it can be used for both. And I think that the more important element of that is that we have not had subcutaneously administered prophylactic agent for hemophilia B, and in fact it closes that unmet need in the context of providing prophylaxis for the hemophilia B patient population. I think one needs to perhaps nuance this in the context of inhibitor versus non-inhibitor. For the patients without inhibitor, the major step from the on-demand treatment, essentially it's a reduction in the treatment load that one is experiencing when you're giving Marstacimab subcutaneously once a week. I think, in fact, it will be a very useful alternative to the current intravenous routine factor prophylaxis in selected patients who clearly would like to have a subcutaneous regimen, who may have poor venous access, and who may have challenges, in fact, with regard to adherence, and who may very well be, I mean, hemophilia is a lifelong condition, maybe having some fatigue in treating hemophilia, particularly in treating hemophilia prophylactically. From the inhibitor perspective, my take on it is that we have had quite suboptimal therapy with bypassing agents in the past. And we can see from the results of the latest basis clinical trial in the inhibitor population that it is possible to prevent bleeds in these patients, which was not the case in the past. So the notion that inhibitor patients will forever carry a very high bleed rate, it's beginning to be changed with these molecules, including, of course, Marstacimab. I think it will be remiss for us to think that, in fact, phase 3 clinical trials, I think, are perfect. The most important remark I would want to make perhaps is to highlight that the basis clinical trial does have strengths, and I think I won't repeat the safety and efficacy that are acceptable, but it also does have limitations, and the limitations, amongst others, will include the fact that it is, of course, an open label study. It's not a randomized control study, which we don't do very often in rare disease space. So one has to keep that in mind. In fact, with that comment in mind, there's no doubt in my mind that the real value added by drugs such as Marstacimab will be seen when we are examining the real world data, when this drug begins to be used by outside the clinical trial. And some of us can't wait to see that data set.